Okay, so thank you very much for the invitation. So I'm responsible of the computational work in the project. And instead of a presentation, I decided I can actually show you the website we're developing and tell you in the same time how it relates to SUDEM and why it's important for the pharmacokinetic prediction of pharmaceuticals. So we're developing a database system that storing a lot of information about pharmaceuticals. And this information can relate to clinical trials. It can relate to the physico-chemical properties of pharmaceuticals. It can relate to their ADME properties. And with the SUDEM project, we also added a layer where we have environmental fate prediction. And recently, as part of the premier project, which is the EU EMEI project, we're joining forces with the University of Exeter to integrate their own database, which is called EcoDrug, that aims to predict the mechanism of action of pharmaceutical. And their goal and our goal in this other project is to predict the possible environmental toxicity of pharmaceutical. So what you have here on the screen, it's I hope it works. It's the interface that we're building, and it has a lot of functionality to search pharmaceuticals, their mechanism of action, their target. So that's directly related to environmental toxicity, for example. We can search, for example, metabolites. I'm going to click and hope it works. It can be slow sometimes. So this is metabolite network we have stored about pharmaceutical. You can see them as a network. And the idea here is for some of them, we have the excreted fraction, and we have also the, let's say, the percentages of the different excreted fraction. It's a network you can play with, and that allows to kind of to follow what's in order to determine the fate of pharmaceuticals in the environment. We need to start what has been excreted. So either it has been poured down the drain or it has been excreted by the patient. And this type of database help to map the data directly to a certain pharmaceutical. So it's key to today's talk. We have also the mechanism of action, and the mechanism of action is I'm not going to click it because it may take too long to download. I'm just going to click on the compound chart. But the mechanism of action approach. Usually it works, but it's lost on a local computer, and this database will be hosted at the Finnish Scientific for scientific computing. So if you click on a compound, you get a chart of the compound, you have description, you have structure, you have physico-chemical property that will later be critical to make prediction about let's say the fate of pharmaceutical, whether they're photolyzed or they're adsorbing. You have ADME properties, and we have the different targets, and that's extracted for different databases. And this one is the KMBL database. You can follow a different measurement. We have the organism also where it has been measured. So some of the measurements have not been measured in human, but they've been measured, let's say, in rat or in fish or in some other species. We have also, if there is data, pharmaceutical data, pharmacological data, we have also access to this data that will help later to the model. So all of the purpose of this data system is to be able to model different parameters. Then we connect to indication, we connect to adverse reaction, and we also connect to adverse to side effect from the label. So the one that was observed during putting the compound in the market. So this is a very large screen. So this first thing I wanted to show you, metabolites. So we're accessing for a bit more than 100 compound metabolites. We're accessing mechanism of action. We also access, now I will come back to the main page because we don't have so much data. We have also environmental data for some of the compounds. If I search, it should work. So yeah, it's a different compound. This is the way it's stored in our database. Again, if you have data, it's there. This one has some environmental impact data. So then you go to an interactive map. For example, here you can see where this compound has been measured in the environment. So that's actually close to the University of Exeter. So that each is a measurement point, and there is a the data is organized is very structured, so we can know, for example, this is a compound, this is the concentration that was measured at this particular site. There's a reference, a date. This is the one that's measured by our collaborators. All this, I think, this across leads, and you can see the river here. So what are we working on this system still? Well, we're working on the mechanism of action prediction. We're working on models to predict degradation, especially in wastewater purification plants. We will work on predicting mechanism of action as part of the Premier project, not suddenly Premier, but that will be integrated into this data system or this platform. And then we're thinking to add layers of data to these maps. So if we get the population and the consumption of medicine, then we can make accurate prediction that are connected to some geolocalization. The prediction can also, we can add some seasonability index, or we can add some temperature index if we cross data from different sources. So, well, this is what I wanted to show you today. So there's a lot of functionalities that I've not presented in this database, but just because the topic, the focus is not the one as sudden, and this is all related to adverse effects. But the idea is we gathering data on pharmaceuticals, also on compounds that are not pharmaceuticals. For example, agrochemicals or some persistent organic pollutants. They are not in this database. They are in another database we're developing, but we want to use them for read across, so to predict the environmental effect of pharmaceutical. We want to use the other.